Okay, hi everyone. So now we are going to go ahead and dive into meiosis. So meiosis has two stages, meiosis 1 and meiosis 2. So we're going to talk about some of the differences between these first, and then we're going to go into each of the stages individually. So meiosis 1, what is the goal here? We want to break up the siblings, aka our homologous chromosomes. So I'm going to go to this picture on the side really quick to help explain this. So if you look at this first cell right here, this is before meiosis 1. We have 1, 2, 3, 4 chromosomes, and you'll notice you have 2 that are bigger and 2 that are smaller. So these 2 big ones are homologous chromosomes, and the 2 small ones are homologous chromosomes. Meiosis 1 happens in that little section right here. And then we go to this stage, which is right after meiosis 1. And you'll notice that each cell got one big guy and one little guy. So in this first cell, it had one blue big one and one red big one. Ignore the different colors for right now. This cell got the big red one, this cell got the big blue one. So they don't travel together anymore. And the same thing with the little ones, one little one went to each cell. You'll also notice that we said that we have 4 chromosomes in this first big cell. Now we have 1, 2, 1, 2. So we went from 4 chromosomes in this cell to 2 chromosomes per cell over here. So we went from 4 to 2, we split in half our number of chromosomes. And that's why meiosis 1 is called reduction division. You go from 2N, or a diploid stage, to N, or a haploid stage. So in humans, this involves going from 46 chromosomes to 23 chromosomes. If you think about it in a diploid cell or a 2N cell, you're going to have 2 chromosome 1s, one from mom, one from dad, 2 chromosomes 2s, one from mom, one from dad, 2 chromosome 3s, one from mom, one from dad, and so on. And during meiosis 2, you just take one of those into each cell. So now each of your cells has 1 chromosome 1 in it, 1 chromosome 2, 1 chromosome 3. And in meiosis 1, you have something called random assortment, where it doesn't matter where your chromosome from mom and where your chromosome from dad go. So for example, if we have our 46 chromosomes that split into 223 cells, the 23 over here don't all have to be from mom, and the 23 over here don't all have to be from dad. You could have 12 from mom and 11 from dad. You could have 2 from mom and 21 from dad. It doesn't matter how it splits up, and that accounts for genetic variation based on what genes are going where. So that's called random assortment. So now let's look at meiosis 2. Meiosis 2 is all about the identical twins, aka the sister chromatids. You'll notice identical is in these little quotation marks here, because if crossing over occurs, they are no longer exactly identical, but they're still sister chromatids. So looking at this blue one right here, for example, each side is a sister chromatid, and usually sister chromatids have the exact same genes, exact same alleles. And you'll notice on the top right here, obviously they are not the exact same. So we still refer to them as sister chromatids. Just keep in mind, if crossing over happened, they're not exactly identical anymore. So looking right here before meiosis 2, we have 2 chromosomes per cell. After meiosis 2, we split those sister chromatids up. And you'll notice in each of these cells, there's still 1, 2, 1, 2, 1, 2, 1, 2, 2 chromosomes per cell. So we went from 2 chromosomes per cell to 2 chromosomes per cell. So this is called equational division because nothing happened to your numbers. We were a haploid when we started, and we were a haploid when we finished. Each cell gets one of the sister chromatids, and we know when they're together, it's 1 chromosome. They split apart, now it's 2 chromosomes. The other thing I want to mention about meiosis 2 is that it is the most similar to mitosis. We know in mitosis, it's all about splitting the sister chromatids up, and it's the same thing in meiosis. And that in mitosis, you start with a 2N cell, you end with a 2N cell. In meiosis, you start with an N cell, you end with an N cell. So you're splitting the information up equally in both mitosis and meiosis, and they're both splitting up sister chromatids. So now we're going to look at meiosis 1 in detail, talk a little bit about crossing over, and then we'll go through meiosis 2. So meiosis 1 first. Luckily for us, it is the exact same stages as mitosis. So we have prophase, metaphase, anaphase, telophase. And the only thing that's a little different is that there's a little 1 after it, and that just denotes it's happening during meiosis 1. If there was 2 little i's after it, which would be prophase 2, for example, that says it's happening in meiosis 2. So prophase 1, we know that the chromosomes are packing, and crossing over happens in this stage as well. It is very, very important that you guys remember that crossing over happens in prophase 1 of meiosis 1. So make sure you have that fact down, and in the next slide, I'm going to draw a little bit about crossing over for you guys. So we're going to put a pin in that. Metaphase 1, you have homologous chromosomes in the middle. Anaphase 1, the homologous chromosomes split. Telophase 1, 2 nuclei form. Each nuclei has half the number of chromosomes, and each chromosome is made of 2 sister chromatids. So let's go ahead and run through this picture on the side. Prophase 1, you'll see you have 2 big guys and 2 little guys. Imagine the pink is the chromosome from mom, and the blue is the chromosome from dad. Metaphase 1, it's your homologous chromosomes that are lined up. And then in anaphase 1, they are going to split apart, and each side gets one of the homologous chromosomes. So then in our last stage right here, before we go to meiosis 2, we started with 4 chromosomes. Now we have 2 chromosomes per cell, and each chromosome has sister chromatids. So now, before we go and talk about meiosis 2, let's go ahead and talk about crossing over really quick. All right. So looking at crossing over, we know that we have chromosome 1 from dad and chromosome 1 from mom. And then during S phase, we go through DNA duplication, which is making sister chromatids. So now we have our sister chromatids. And you're going to notice that for the middle guys right here, they're kind of touching, they're introducing themselves to each other, that type of stuff. But these on the outside have no interaction with anything. They don't know who these other guys are. They don't see them. They don't know they exist. So these guys that are talking and introducing themselves, you can imagine they're shaking hands, exchanging names, and they're going to exchange some information. So after crossing over happens, this is what it's going to look like. Your chromosome right here on the outside remains exactly the same because it didn't shake hands with anybody. But the one on the middle that did shake hands is going to keep a little piece of that other chromosome. And your chromosome from mom, the one on the outside, stays exactly the same. It didn't shake hands or exchange information with anyone. And the chromosome in the middle did exchange information, and it swapped a piece. So now you'll see that we had crossing over happen. Crossing over is really important in increasing genetic variation. I'm going to talk about why. So if we were to think about this chromosome right here, we're going to say that on the top was brown hair and on the bottom was brown eyes. And for this chromosome right here, we're going to say on the top was blonde hair and on the bottom was blue eyes. Now we flipped the top. So now this chromosome over here has the blonde hair from mom, but still the brown eyes that were on the bottom. And our new mom's chromosome has brown hair from dad and blue eyes on the bottom. So what you'll notice is that we kind of flipped what went together. And so now you have different combinations, different probabilities, and that's where the brown hair, blue eyes, brown eyes. And now we have blonde hair with brown eyes, brown hair with blue eyes. So that's genetic variation. And I'm going to briefly talk about the probability of this. This is really important in chapter 7. So just kind of keep it in the back of your head. How many DNA molecules are, let me get my pointer out, right here. there's 1, 2, 3, 4 DNA molecules. For DNA molecules, just count the sticks. How many of those DNA molecules were affected by crossing over? You should notice that 2 out of the 4 were affected. Oh, I ran out of room. That's a 4. 2 out of the 4 were affected by crossing over, which is 50%. So if crossing over were to happen every single time, we would only actually affect 50% of our chromosomes, because these two on the outside, right here and right here, are never going to change because they don't have any interactions. It's only those two in the middle that have interactions. So only 50% of our chromosomes, if crossing over happens every single time, would be affected by crossing over. All right. Let's look at meiosis 2. So again, this is really similar to mitosis. Everything just has the two like eyes after it for prophase 2, metaphase 2, anaphase 2, telophase 2. Prophase 2, pack the chromosomes. Metaphase 2, sister chromatids are in the middle. Anaphase 2, you split apart the sister chromatids, and telophase 2, you split into 2 nuclei. So what you'll notice here is that the big difference between meiosis 1 and meiosis 2 is if you're dealing with homologous chromosomes or sister chromatids. If we went back to meiosis 1, you'll notice everything was about homologous chromosomes. Metaphase 1, homologous chromosomes are in the middle. Anaphase 1, homologous chromosomes split. Meiosis 2, it's your sister chromatids in the middle. Sister chromatids split. So that's a really easy way to tell them apart. If you ever see homologous chromosomes lined up, you know you're in meiosis 1. All right, so that was the end of what I had for you guys for meiosis. Oh, I guess one more comment really quick to talk about the genetic results is that you have 4 cells. 4 cells, and they are not identical to the parent cells, obviously, because the parent cells, these aren't the parent cells. That was the end of meiosis 2, but if you went end of meiosis 1, sorry. If you went all the way to the very beginning, it would have had 4 chromosomes to start. So these are not identical to the parents. There's 4, they're haploids, and they're genetically different from each other. So you notice this is like big blue stick, small pink stick. And then over here, big pink stick, small blue stick. And these are all mixed up. So they're genetically variable. There's 4, they're haploids, they're not identical to the parent. Mitosis, there's 2, they're diploids, they're identical to each other and the parent. All right, and that's all I have for you guys for meiosis. And the last tricky topic video is going to be over chromosome counting.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.